Hey guys, welcome to the BioLofty channel. It's your host here, Mr. Loftman, Mr. Goat to some of you. I know it's been a while since I posted my first video, but I am going to start trying to post all of our lectures online here. So if you guys are part of my class, you can obviously watch them, get some extra feedback. If you miss class, I'll be here for you to see. And if you're not part of my class, welcome. You're more than welcome to watch these videos and learn a little bit about biology. Should help. Right, so getting right into it. Mutations. Talked about them a little bit in class. Let's go over them again real quick. So these are all mutations. Anything from a seedless watermelon here, as benign as that may be, and the blue-eyed baby, relatively normal mutations that won't affect the organism that much. They can also be pretty detrimental, such as the mutation here with the fingers coming together, the kid with no arm right there, and the legs that seem to have some defects. Those can be mutations as well. So there's a bunch of different mutations, and we're going to get into them during this lecture. So what are mutations? Mutations are any change in a DNA sequence. Normally when people think about mutations, they're really thinking about Wolverine right here, right? X-Men, superpowers, all that stuff, or they're thinking something like terribly wrong that causes a disease in an organism. Now that isn't always the case. Mutation can just be any change in the DNA sequence of an organism. It could literally be changing an A to a C, something as simple as that. So are mutations good or bad? Both, actually. It depends on what the mutation is. So they can affect your genetic material, which can lead to cancer or genetic diseases, obviously. If things go wrong with mutations, changing your DNA, it can cause some terrible, terrible mistakes. But at the same time, there are good mutations, such as seedless fruits that we eat all the time. We have selectively bred them, so we get that mutation to show up more, that ends up having no seeds. And we don't like eating seeds. So, all part of the plan, right? Okay, so what types of mutations are there? There are gene mutations and chromosomal mutations. There are others, but those are the two big ones that we're kind of going to focus on right now. And as you can probably imagine, gene mutations are just going to be on the gene level, affecting one or multiple genes, while chromosomal mutations are affecting multiple chromosomes. So, what are the types of gene mutations? Point mutations are one of the types, and these, compared to the other type of gene mutation, are usually better. So, a nonsense mutation is when an amino acid, or changes in the DNA sequence, cause an amino acid to change to a stop codon. You can see that right here. We have this ATG at the beginning over here, and it just makes a tyrosine. But then there's a mutation that changes that G to a C. And when the G is changed to that C, it becomes UAG, and it becomes a stop codon. Now, we call that nonsense because it doesn't make any sense, right? So you're coding for this gene, you're coding for this protein with this gene, and you're just going along, and all of a sudden you hit a stop codon, and you just stop? That doesn't make any sense. That's how I remember it anyway. So nonsense mutation just changes an amino acid to a stop, and it stops synthesis right there. Missense. So missense is actually changing one amino acid in the sequence, but the gene is continued to be read as normal. It doesn't get stopped. An example of this is right here. So we have a changed to a G, which is going to change ATG to GTG, which is going to make histidine instead of tyrosine. So in that case, it depends on the protein. Sometimes you can change one amino acid for another, and it doesn't really do anything. Other times it can cause huge disastrous side effects. An example of a missense mutation is sickle cell anemia. So the sickle cell disease is also a missense mutation. It's just a change in one letter that ends up changing how the entire kind of molecule works inside the red blood cell, and it can cause a lot of issues. Now we also have silent mutations. It's the last type of gene mutation, or the last type of point mutation, I should say. And silent mutation usually affects the last letter of a codon. So as you can see here, it's ATG, and it's changed to ATA. And basically what's happening in a silent mutation is you're still getting the same amino acid out. Which is why it usually takes place in the last letter of the codon, because that's the one that you can often change a little bit and get the same amino acid. So it's called a silent because it doesn't really affect anything. You have the same amino acid coming out as part of the protein at the end. So there's really not a huge selection pressure to get rid of it. You a lot of times don't even know that it's there. Alright, so some other types of gene mutations in one sec. Examples of point mutations again. This top one, we're changing a CAA to a CAG. And as you can see, we're both getting the same amino acid out of both of them, so that's going to actually be a silent mutation. This one's a UCA to a CCU, or sorry, a UCU to a CCU, and we're changing a threamine to a proline. So what's happening there is that's actually going to be a missense mutation, where we're getting a different amino acid out at the end. And this one right here, UAU to UAA, that's tyrosine to stop. And that is a nonsense mutation because it doesn't make any sense. We're just stopping right there. Alright, other gene mutations we're going to cover real quick. Frame shift mutations. These are the nasty guys. These will mess you up a lot of times because what happens is in those last mutations we were looking at, we were only changing one letter out of your entire gene, which can a lot of times be kind of overlooked or only change one small part of the protein. This is going to mess everything after it up. The reason for that is we have deletions and insertions, right? So as you might be able to imagine, a deletion is where we get rid of a base in a gene, and an insertion is where we add an extra base in a gene. So these are all insertions. You can see right here, CAG, CCC, ACT. So we're throwing a T in between this G and C right here. When we do that, it doesn't only mess up this one codon like the last one does, it messes up every one after it. So the proline's getting changed to a serine, the threonine's being changed to a histidine, and it's going to keep going on and on for the rest of the gene. That's why these are really, really bad. Now there is an exception. You can get a 3-codon insertion, or sorry, a 3 nucleotide insertion, and that'll just add another codon. That isn't as bad because the ones after it are going to keep working. It can still mess up the protein. Some examples really quick. So here's our normal gene on top right here, right? As you can see right here, we inserted this extra A for the insertion mutation. And for the deletion mutation, I put this in parentheses because we deleted a G that used to be there. It's not there anymore. So you should pause the video right now if you want to practice. And I want you to draw the lines where the codons are going to be so you can see for yourself. And now I'm going to show you guys where they're at. So here are where the codons are going to be for the first gene. So real nice, just 3 nucleotide codons, just looks pretty normal. That's how it's supposed to be. Now in this one, okay, it looks good, looks good, looks good in insertion so far. And then we get here. And as you can see, that's going to be a different amino acid, or maybe the same. I didn't actually check if GAT and GAA make the same amino acid. But everyone after that is going to be off. So instead of the next one being a GTC, it's a TGT. And then instead of CGC, it's a CCG. So every one after that in the gene is going to be incorrect. Now for deletion, it's going to be pretty much the same thing. So we have that one's normal still, and then almost immediately it's going to be messed up right there because I put it near the beginning. So as you can see, getting rid of that deletion, that's why there's 4 letters here. I'm not counting it because I deleted it. It's not there anymore. I was just showing you guys which one I deleted. So now instead of a GGC, it's going to be a GCT, and every single one after that's going to be messed up too. And that's why frame shift mutations are so incredibly dangerous compared to point mutations, because they change so much more. Alright, we're going to real quick go through this stuff. It's not quite as important as the other stuff, but it's still very important for you to just have the knowledge. So chromosomal mutations are when chromosomes are broken or lost during mitosis or meiosis. This is common in plants. Actually, it's how plants evolve a lot of times. So I know we did that strawberry lab in class. So strawberries actually have 8 copies of every chromosome. And that actually happened via chromosomal mutations accumulating in them over time. But it can cause non-destruction in other organisms, which can cause various diseases such as Down syndrome in humans. So this is a chromosomal or gene mutation. This is a gene mutation like we talked about. That is a missense, right? And what causes mutations real quick? So a lot of things can cause mutations, but basically DNA damage from environmental agents such as UV light, nuclear radiation, or certain chemicals. Mistakes that occur when the cell copies its DNA in preparation for cell division. So again, all mutation really is when the cell's dividing and it's copying the DNA, it makes a mistake. And it's more likely to make a mistake if the cellular machinery that's copying the DNA is damaged or if the DNA itself is damaged, which can be damaged by various things such as smoking, ultraviolet light, radiation, all the stuff that you normally hear to kind of stay away from, right? And there's a reason for that. Alright, so that's all for this video. I hope you guys enjoyed it. I'll be sure to keep updating these from now on and posting these all online, and I will see you all soon. Thanks again for watching as always. We appreciate your stewardship.